I would just like to thank the OIS organizers, especially Craig and Maureen, dear Maureen, wherever you are. Thank you so much for giving us the opportunity to present today. Aramis Biosciences, we are currently developing a topical immunomodulatory treatment for dry eye disease. The company was founded based on the research of Dr. Reza Dana and Sunil Chauhan, who are out of Mass Eye and Ear. Dr. Dana and Dr. Chauhan identified that T helper 17 cells, which produce IL-17, are the primary effector cells in the pathogenesis of dry eye disease. Armed with this insight, the team then set about looking to develop a small molecule, really as an alternative to a biologic, which will be more suitable for delivery to the ocular surface. We actually ended up partnering with Dompe Pharmaceuticals out of Italy. And with Dompe, we used their artificial intelligence drug discovery platform, and we developed A197. A197 is a novel first-in-class IL-17A antagonist. In Q4 of 2021, we actually closed our Series A round of financing, and thanks to our support from our investors, Santon Ventures and Sapphire Partners, we were shortly thereafter able to file our IND and then commence our first proof of concept study in dry eye patients. Dryness on the ocular surface basically leads, as you all know, to epithelial cell damage. As a result of this, you get activation of immune cells which migrate to the draining lymph nodes. In the draining lymph nodes there, these cells present and basically to naive T cells which differentiate Th17 cells which migrate back to the ocular surface where they release IL-17 which interacts with the IL-17A receptors and basically furthers the inflammatory process. Another aspect of this which is important to recognize as well, in the lymphoid tissue, Th17 cells suppress Treg function. And it is important that in order to correct, you need to correct Treg function in order to break the vicious cycle of inflammation. While IL-17 is a novel target in dry eye disease, it's important to recognize that it's extremely well known in other immune-mediated conditions, in particular in psoriasis. And actually, as you look at the history of drug development in psoriasis, it's actually quite remarkable some of the parallels you see with dry eye disease. And Paul touched upon this a little earlier. In days gone by, people in psoriasis, patients were sort of poorly managed with creams, potions, looking at steroids, immunosuppressants. But it was in 2002, with the advent and introduction of the biologics, you started to see change. And interesting, the first biologic that was introduced was Raptiva. Raptiva was an LFA1 antagonist, which is, of course, the same mechanism of action as lifitegrast. Fast forwarding to today, the gold standard of treatment for psoriasis are the IL-17 inhibitors, Cosentyx and Taltz, and they truly have revolutionized the treatment of psoriasis. In many patients now, you're getting almost complete clearance of plaque in psoriasis. Paul touched upon this, and you've obviously all seen this before, that there's a huge unmet need in the dry eye arena. As Paul mentioned, there's about 18 million patients who've been diagnosed, and fewer than 1 million patients are actually receiving an FDA-approved product. And you have to wonder, why is that? And part of the reason may be that some of the treatments that are currently available have tolerability issues and also perhaps don't have the level of efficacy that patients might expect. Looking at prescription data from 2022, 90% of the prescriptions that were written for the two key products that are there, Xiidra and Restasis. But what's really more interesting when you look at this is that 40% of those prescriptions were actually for new patients. So what is happening is there is a large number of patients that are not getting their prescriptions refilled. So we have a $1 billion plus category that, because of the prevalence of the population, what really is happening is that the market is being maintained by the funnel being filled by new patients as opposed to patients actually receiving refilled prescriptions. As I mentioned, we recently completed our phase 2A proof of concept study. This was our first in human experience. It was a multi-center, double-masked, randomized, vehicle-controlled study in 207 patients with dry eye disease. On the left-hand side there, you'll see some of the key eligibility criteria that we used for the study. The key objectives for the study were, first of all, since it was our first study, was to evaluate the safety and tolerability of A197. We also wanted to confirm biological activity, both in terms of signs and symptoms. We also looked at dose response. We actually included in this study a Xiidra arm as well. We wanted to see how our product would stack up against Xiidra, both in terms of tolerability, but also in terms of efficacy. And then finally, we wanted to identify what we would, the most appropriate population here for us to actually look at in future studies. At a high level, the results of the study were extremely positive. We demonstrate very, very good tolerability with A197. From an efficacy point of view, our high dose of 0.1% demonstrated a very rapid onset of action and also a durability of effect. And we actually saw that the most marked separation, both from vehicle and Xiidra, at day 28. And finally, we identified a subgroup of patients who we believe would be prime candidates for our next study. So we see a pathway forward for approval for the program. In terms of tolerability, A197 0.1% was very well tolerated. As you'll see here on the slide, 12.3% of patients reported an ocular adverse event, as compared to 11.7% of patients who were on vehicle. If you actually look at these adverse events and see if they were deemed related to treatment, that number drops from 12% down to 7%. In comparison, 32% of patients who received Xiidra reported an ocular adverse event. In terms of, so we actually looked at we had a pre-specified subgroup of patients in this study. This group were patients who at baseline had a total corneal fluorescein staining score of less than 12 and an eye dryness score of greater than 60. And when you look at this subgroup, the patients who received 0.1% of A197, they showed a 0.71 or a 27% reduction in inferior corneal fluorescein staining at day 28. This compares to a 7% change on vehicle and 8% on Xiidra. As I mentioned earlier, this study was not powered for statistical significance, but rather to guide us for future development. So we've actually just looked at some sample size estimates, and based on this data for future studies, we believe that 200 patients or 100 patients per arm would give us 90% power to show a statistical difference both against vehicle or against Xiidra. In terms of symptom, again, similarly, the 0.1% dose, again, in the same subgroup analysis, we showed a 23% reduction in symptom score as measured on a visual analog scale, or a 33% change, compared to 12% on vehicle and 8% on Xiidra at day 28. Again, as per the previous slide here, if we look at sample size calculations, again, a 200-patient study would actually give us significant power to show a difference both between either against Xiidra or against vehicle. So in terms of next steps, having confirmed safety and efficacy in our first in-human proof of concept study, our plan next is to conduct a phase 2/3 clinical trial evaluating A197 for a treatment duration of 28 days to the rapid onset of action. Additionally, we're also pursuing a $25 million Series B financing to advance the program, not only to conduct this next clinical study, but also to complete the other non-clinical work that needs to be done. So in summary, at Aramis Biosciences, we now have a first-in-class new chemical entity with a novel mechanism of action targeting the immunopathogenesis of dry eye disease. We have achieved proof of concept. A197 was extremely well tolerated, which we think is extremely important for new products within the dry eye arena. It was also had a rapid onset of effect, which again, we believe patients want to see. and we have identified a clear pathway forward to demonstrating superiority either against vehicle or Xiidra in future pivotal studies. Thank you so much.